As people get used to the new normal, can our behaviours and experiences over the past year help shape and improve how we live?
Wouldn't that be wonderful?
Irish research continues to make advances in human health and the health of our planet.
That's so amazing.
This week on 10 Things to Know About, we're checking out blood. Jonathan and Kathriona meet the researchers investigating the risk of blood clotting in COVID-19 patients, and for something totally different.
I'll be exploring efforts of Irish researchers to turn blood into a completely biodegradable and nutritious plastic.
Blood is literally our life force, transporting oxygen and nutrients to our organs, removing harmful carbon dioxide, and fighting off everyday infections. But this can all be thrown out of kilter by an outside actor, like a virus disrupting that delicate balance. As COVID-19 spread across the world, doctors and scientists were scrambling desperately to try and understand this new disease and the devastating impact it was having on patients. While most of us are familiar with the respiratory-like symptoms, many of us are unaware of the hidden dangers of this virus in our blood. I'm at UCD in Dublin to meet with a team of researchers who have been looking at the effects of COVID-19 on the platelets in our blood. Patricia, what are blood platelets and what do they do?
When you cut yourself, Jonathan, what platelets do is they get to the site of injury first. They're like our little first responders in our blood. So they get there and then they just all come together and they seal the gap. And when they swim around in our blood for their 10-day lifespan, they pick up information in real time, but they also send out information and they send it back to our bone marrow, which makes our blood, and that kind of tells the bone marrow to make a slightly different quality of blood cell to put out into our blood.
Do they cause blood clots in the body then?
Yeah, so the platelets are swimming on the outside of the blood, and that's really close to cells called endothelial cells, which line all our blood vessels. When they're circulating around, they're constantly getting signals from our endothelial cells to keep them quiet. So we have these suppression mechanisms to keep our platelets sleeping. You know, nature does not want us to bleed to death, okay? So when you cut yourself, platelets need to react quickly because you want a clot within like 30, 60 seconds, okay? And so platelets are primed to come and form that clot and all come together to form a clot to seal the gap.
Is that what we're seeing in COVID, this failure to suppress the platelets coming together?
Yes, it really is, and it actually seems to be quite acute.
Patricia is working with Dr. Barry Kavan, a consultant haematologist at the Mater Hospital.
We know our usual practice that the vast majority of blood clots which occur in hospital are potentially preventable with standard low doses of anti-clotting medicines. But what became apparent all over the world was that patients admitted to hospital with COVID had very high rates of blood clots despite these usual standard measures. And this is incredibly important because a blood clotting event is a potentially fatal event. And in some of the reports which we've seen from around the world, the risk of developing a blood clot in some high-risk populations of COVID patients was reaching 25 or 30%. So this is obviously a huge concern to us here and has huge implications for our practice, but all over the world this has sparked a huge amount of research interest from a clinical and scientific perspective, aiming to try to, I guess, characterize the molecular mechanisms which are underlying this very unusual link between severe COVID and blood clotting, but also to try to develop new treatment strategies and new preventative strategies to help improve outcomes for patients and reduce the risk of these potentially fatal events.
Patricia and her team are analysing blood samples from COVID patients to look for the evidence of biomarkers, signals in the blood that could help predict whether somebody who goes into hospital and tests positive for COVID-19 might go on to need intensive care.
What we're able to do here is we're able to look for a particular protein that tells us how activated the platelets are as they circulate around the blood. So Anna will add in a buffer on top and you will see a colour change happening, and the more blue that it changes colour to is the more of that protein that's there. So you can see now that it's actually turning blue.
Oh, yeah. And so we would see this quite a dark blue in severe COVID patients.
That's what we found, Jonathan. Shane is looking at anonymised data that's like what we looked at to develop our COVID-19 algorithm. So we looked at blood test data from all the patients with COVID-19 that went into the Mater Hospital during the first lockdown, and we were able to take all that data and use artificial intelligence to look at that data in a very non-biased way to see is there any particular markers in there that will tell us, be able to predict the future, basically, be able to tell us how that patient's COVID will progress.
To date, the researchers have carried out a successful pilot study of their algorithm, and the next phase will involve full hospital trials. As we continue to live with the effects of COVID-19, the team's aim is to see their technology implemented into hospitals in the next couple of years. That's really exciting. So it sounds like if you figure out the differences between the sort of information, you'll be able to see which COVID patient will likely need ICU, which patient would likely go home.
Our hope is to really be able to help understand COVID-19 better, why some people's platelets are more active than others in COVID. What we really want to be able to do is help doctors make better decisions when they have a COVID-19 patient sitting in front of them.
Blood clots can form in many different parts of the body, but Irish scientists have discovered unusual microclots in the lungs of some COVID-19 patients. Hi, Jamie. Jamie O'Sullivan is from the Royal College of Surgeons in Ireland. Tell me, how did the understanding of the clotting aspect of COVID evolve?
So I suppose the early picture of COVID really came out of Wuhan in China, where they were seeing lots of abnormal clotting in their patients with, I suppose at this point, very little understanding of why. And then as it kind of spread as a wave through Europe, there were a number of autopsy reports done, a limited number, that being said. And what they found in the lungs of these patients post-mortem, unfortunately, was that there was a widespread deposition of these microclots within the lungs. And we think that might be a consequence of the way that COVID probably entered the body, which was via the lungs.
And is that why people then have symptoms of breathlessness and coughs and that kind of thing?
Yeah, exactly. So what happens is when you develop these microclots, which in the vessels that line the lungs, it causes almost like congestion of those vessels, and it impairs oxygen to flow in and out of the lungs as effectively as it normally would.
And that's because in our lungs we have a huge amount, like obviously the blood oxygen exchange thing that's going on, there's this big concentration of blood vessels.
Exactly. There's about 300 million of these small capillaries, as they're called, or micro blood vessels within, lining kind of around the lungs, really key in promoting that oxygen exchange and allowing us to breathe. And when you start having all these microclots getting deposited within these capillaries or small blood vessels, it really blocks them up. The microclots themselves are typically limited to severe hospitalized COVID-19 patients. But what's really interesting is that patients with long COVID syndrome have elevated markers of blood clotting in their blood that we take from them maybe 10 to 12 weeks after they've had the initial acute COVID illness. So while they might not have clots, we think that they might be at increased risk for clot development many, many weeks after they've had long COVID.
Our knowledge of long COVID is evolving all the time, but it's estimated about 10 to 20% of patients that have had acute COVID may develop long COVID syndrome many weeks or even months after they have had the initial illness.
The common symptoms that are reported are things like fatigue, shortness of breath, even heart palpitations, but it ranges quite variably. And we have looked in blood samples from these patients who've attended a post-COVID clinic to see what's going on. And what we see is really high levels of pro-clotting factors in their blood many weeks later. And these patients with the high levels of these pro-clotting factors seem to have more fatigue and reduced physical fitness. And we think that maybe this clotting activation may be at least contributing to some of the root cause for long COVID.
So you're trying to unpick, is it to do with the blood vessels? Is it to do with the blood itself? Is it to do with the inflammation response to the thing? You're trying to unpick what is causing it.
Yeah, exactly.
So should long COVID patients be put on blood thinners?
Yeah, it's a really interesting point, whether these patients may benefit from being on anticoagulants as we know them, which would thin the blood and potentially prevent life-threatening clotting complications. But it's really important to appreciate that these blood thinners carry a risk of bleeding that isn't insignificant. So it's a really delicate balance in managing these patients and trying to decide who is at high risk and would they benefit. And there's a lot of ongoing clinical trials globally to try and work out the optimum dose for blood thinners and which patient groups would benefit from this. We're really only beginning to understand long COVID syndrome. I think a conservative estimate might be between 60 to 80,000 people may present or have presented with some form of post-COVID or long COVID syndrome. And I think the more that research can tell us about the root cause of long COVID, then we can begin to develop targeted and effective treatment for these patients.
So if you are one of the potentially 80,000 people who are suffering from long COVID, you should take a little bit of heart that science is on it. You're trying to unpick it all and get to the bottom of it.
Yeah, I think COVID is such a good example of scientists and researchers and clinicians collaborating across the globe to really, bring forward information and provide insight into this, new disease at such a rapid pace. And I think the vaccine program has really been testament to that.
It's 1897. A little-known Irishman is about to write both himself and one of literature's most famous fear-inducing characters into history. Bram Stoker's Dracula spawned a genre of writing, a fascination with vampires, and a newfound respect for garlic. Yet amidst all the supernatural chills, the book was the first work of popular fiction to feature one of science and medicine's most important breakthroughs, the blood transfusion. At the time of writing, blood transfusions were a poorly understood procedure, with patients frequently ending up dying due to blood complications, and doctors, with their limited knowledge of blood, being none the wiser. But a great novel never lets truth get in the way of a good story. So, after being bitten by Dracula and falling victim to vampire-induced anemia, one of the novel's main characters, Lucy, receives numerous blood transfusions, which in good news returns her vigor, but in bad news doesn't stop her slow decline into becoming a vampire. Leaving Stoker's Transylvania behind and fast-forwarding just 3 years to 1900, vampires' thirst for blood had been matched by scientists' thirst for knowledge, with the discovery of A, B, and O blood types, which revolutionized transfusions and allowed doctors to predict which blood types could be safely mixed. So remember, if interviewed with or bitten by a vampire, a blood transfusion might save your life, but it won't stop you joining the ranks of the neck-biting, blood-sucking undead. See you next time.
Approximately 3 to 4% of a pig or cow's body weight is blood. Now that mightn't sound like a lot, but it's about 3.5 litres for a pig and 18 litres for a cow. In order to limit waste, scientists are looking at novel ways of turning blood into high-value products, creating a more sustainable industry. In terms of global challenges, reducing our plastic waste and increasing our agricultural sustainability are both extremely important. So developing solutions to address both issues at the same time is a no-brainer. Anne-Maria Mullen is overseeing a research programme that aims to make the most of our meat. Anne-Maria, when we're producing meat, how much of the animal actually ends up as meat and how much is waste?
About 40% of the animal is meat, and 60% is other. I would avoid calling it waste because very little is actually wasted. The industry have been very dynamic at getting markets for a lot of this product, but a lot of it is still either not going to an edible market, so it's not been used for human consumption, or it's not getting very high value.
If the meat is definitely one thing, are they actively looking for things to do with the other 60% of the animal?
At the moment, most of the blood would be categorised as an animal byproduct, so that has a particular connotation in terms of EU regulation. And if it's an animal byproduct, it's not fit for human consumption. But it can be collected in a way that allows it to be used for human consumption. And that's one of the principles in the Novel HemePack project, is can we find better ways to use our blood where the blood is separated into what we call a cellular fraction, which is the red blood cells essentially, and then plasma or serum depending on how it's prepared.
One of the most intriguing aspects of Anne-Maria's research is turning this blood byproduct into a completely biodegradable plastic that could be used for food packaging. How can blood then be used to make a plastic?
So yeah, we use hemoglobin protein as one of the main components in producing the bioplastic, and it's combined with other ingredients to produce what we would consider a good bioplastic that we would hope would be comparable with a petroleum-based product for certain applications.
Many plastics in use today can be degraded or broken down, but that often takes years and needs complex industrial processes to make it happen. Instead, bioplastics can degrade in soil within a few weeks. Carlos Alvarez is a member of the research team that is developing bioplastic film made from pig's blood.
When we want to design this film, we need to compare this with the plastics that are already used in the market, so the petrol-based plastics. If you need to compete with them, your mechanical properties need to be equal or at least similar to that in the market already. The process takes us several steps until you get the final product. So the first one we do is to collect the blood in the slaughterhouse in an hygienical way. It means that the blood is collected in a closed system, so there is no risk for contamination. Then to the blood we add some anticoagulants, so it prevents the blood to get clogged. You separate the blood in two different fractions. One is the liquid fraction, which is the plasma, and the other one is the solid fraction, which is composed by the red cells and the white cells in the blood. What we do next is we dry these liquid fractions and the solid fractions using freeze dryer machine, so we get a final powder. So what we have in here, this is the red cells after being fractionated from the blood and dried. We dissolve this in water and we get this type of solving solution. And we add some plasticizer, which it helps to the proteins to actually form the plastic. So it gives the flexibility to our plastic. After we do that, we pour our solution into this type of plastic molds.
Okay.
And then once we have our solution in here, we put this into an oven at 35, 36 degrees. Once they are dry, what you get is the blood films, okay? So you can see they are solid, they are flexible, but the color at this moment?
Yeah. Do I want to see that color wrapping a
Probably
food product?
not. We realize as well that when you put these films in contact with water, they almost disappear. They start to disintegrate. So we did several trials trying to modify the color and the mechanical properties of the film and also to reduce the solubility. I just wanted to show you one of the trials we did. As you can see, this is full of bubbles, it's not consistent. That was one of the many that didn't work. But after optimizing the process, what we got at the end are these type of films. As you can see, are flexible as well, but they are much more resistant to the mechanical stress. The color is much nicer, so it's almost transparent.
So it must have been really frustrating going through all of the different versions.
Yeah, science usually is trial, but also you need to learn something from your own mistakes. And yeah, it ended up with this product.
I love that you're learning from every failure and you get to something that looks like any other plastic, but it's probably biodegradable. It's definitely better for the environment. As the team develop each new bioplastic film, Carlos subjects samples to a series of strength tests to find out how much force they can withstand before breaking. This characteristic will be a key driver of any successful product packaging reaching our supermarket shelves. And then the next stage is all about upscaling the technology.
1 liter of blood, we can make maybe 40, 50 films of 20 by 20 centimeters each. And at the moment, we're doing our films one by one in a batch base, but we want to do this in a continuous base. So we wanted to generate larger films, so the films are generated continuously in a working day, let's say in a factory.
And how long do you think it will be before something like this comes to the market?
We have seen some previous projects in these areas, and they were able to do a continuous system. So we are hoping that we can adapt this system, but probably it will take 3 or 4 years until we get this point.
We have focused a lot on the science and technology and getting that right, but the other side of it is the marketplace. The petroleum-based plastics are long established, and definitely breaking into that is difficult. Cost is one of the big challenges, but at the same time we've policy and legislation coming down that's forcing the move towards non-petroleum plastics. So something has to give to allow bioplastics to become more successful.
What kind of products do you think it will be used for? Will it be used in the foods that you and I eat, or what are the ideas that are there at the moment?
The bioplastic we're currently working on is from a porcine blood. So, the applications for that would be in a pork context, or possibly beef context as well. But there are many non-food applications that could be considered, whether it's plastics that are used on farms, packaging plastics. So it would depend a bit on the ultimate properties of the plastic when we have it optimized as well as to what is the best application for it.
Wouldn't that be wonderful, though, if the plastics that we were using on the farm, for example, like storing the farm feeds, if that was biodegradable?
Absolutely.
I mean, wouldn't that be wonderful for the sustainability of the country?
It would. It would be a huge impact in reducing the plastics that have to be disposed of.
Do you think people might be squeamish about using something that has come originally from blood?
It's important to be upfront and inform the consumer about what it is that we're doing and why. But we do rely on meat as a food source, so it's kind of our moral obligation to make sure that we're using everything from the animal in the best way possible. And if we can do that in a way that's also reducing the reliance on petroleum plastics, then that's a win-win in my view, that we're getting the nutrition side of it right, and then we're also trying to improve our environment as well.
That's our 10 things to know about blood. Next week, we hit the coast and meet the researchers helping to preserve the biodiversity of our beautiful seaweed forests.
How beautiful is that?